Good morning. Thank you to the EPA, HSE and DSRI for giving me the opportunity to present on the AREST project this morning. My name is Professor Dearbháile Morris and I'm from the National University of Ireland, Galway. The AREST project is a 4-year project funded by the EPA and HSE. As we all know, antimicrobial resistance is a One Health challenge. The One Health approach recognises that the health of humans, the health of animals, and the health of the environment we share are interlinked, and there is a need to bring together the different sectors to effectively tackle the challenge of antimicrobial resistance. Antimicrobial resistance is recognised globally as one of the most important human health challenges we're facing in today's world, and reached the floor of the UN General Assembly in 2016, only the 4th time in history that a health item has been brought to that forum. Following publication of the World Health Organization's Global Action Plan on Antimicrobial Resistance, a number of governments throughout the world have published their own action plans, including the publication of Ireland's National Action Plan on Antimicrobial Resistance in October 2017. And the findings of the AREST project will inform research gaps for the development of INAP2. It is recognised in revisions of all of the national action plans published to date that there is a need to better understand the role of the environment in the transmission and persistence of antimicrobial resistance. As we know, antimicrobial resistance arises because we use a lot of antibiotics, both in human and veterinary medicine, as you will have heard in the presentations earlier, and sometimes not always appropriately. And there is potential for these antimicrobials and antimicrobial resistant bacteria, if present, to enter the environment. It was great to see the publication of Ireland's first One Health report, which brought together antimicrobial use and antimicrobial resistance data in humans and animals. This was published in January 2019, and it is good to know that environmental data will be included in further revisions of this report, including data generated by the AREST project. So when an animal or a human takes an antimicrobial, not all of the antimicrobial will be metabolized within the body. Some of it can potentially enter the environment in a largely biologically active state. If there's antimicrobial resistant bacteria present in the gut of those organisms, be they humans or animals, that can also enter the environment in the waste of that animal. Generally, human waste is treated before it's emitted to the environment, but as we know from the most recent EPA Urban Wastewater Report, there are still 36 locations around Ireland where raw sewage is being emitted into recreational waters in an untreated state. As we know, land spreading is an important part of the circular economy, but again, if there's antimicrobials or antimicrobial resistant bacteria present in the waste of animals, this will be spread onto agricultural land, largely in an untreated state. So there is potential for these organisms and antimicrobials to enter the groundwaters, enter our surface waters, and if these act as sources of our drinking water supplies or are used for recreation or irrigation of food supplies, there's potential for transmission to humans. We know, and I don't need to tell this audience this, that dispensing raw sewage in the vicinity of recreational bathing waters is a risk for transmission of antimicrobial resistant organisms. And we demonstrated this for the first time in Europe through the detection of CPE in seawaters, which was published in 2017. We know the current wastewater treatment processes based on previous EPA-funded research does not remove all antimicrobial resistant organisms. We know that land spreading of organic wastes, both human, animal manure and sewage sludge, is an important part of the circular economy. But based on previous EPA-funded research, concern was expressed by the authors of this report about the presence of emerging contaminants, including antimicrobial agents, in biosolids that may lead to a buildup in soil and enter the food supply. Studies have shown indeed that land spreading of manures increases the proliferation of antimicrobial resistant organisms and the abundance of resistance genes in soils. So we need to better understand what the level of resistant organisms is in manures from different sources, what treatment is effective at removing them, and what application on soil has. So the AREST project is very much taking a One Health approach to understanding antimicrobial resistance and the role of the environment. It's bringing together both national and international experts in human health, animal health, and the environment. And we will carry out, we are in the process of completing 4 integrated tasks. to map hotspots of the drivers of antimicrobial resistance in selected local authority areas, namely Galway City Council and Galway County Council, Fingal County Council and Cork County Council, to assess the relative contributions of various sectors, including healthcare and agriculture, to antimicrobial resistance in the organisms, assess the efficiencies of treatment processes for the removal from drinking waters, wastewaters and manures, and we aim to develop a risk ranking protocol to assess the relative contribution of the various sectors and the sources and levels of resistant organisms in the environment. So, and the first step in this process, and what I will do through the rest of this presentation, is briefly outline some of the activities and the findings of the AREST project to date. The first step was to understand better the role of the natural aquatic environment, and we completed a scoping review on this, which was published in Water Research earlier this year. We also completed a mapping process to help guide the sample location. So again, we were focusing in particular in this project on Galway City and, and County Council local authority areas, Fingal County Council and Cork County Council. Just briefly to take Cork County Council as an example, we mapped the potential sources of antimicrobial resistance. So we mapped the locations of hospitals, long-term care facilities, areas of intensive agriculture. We could then overlay this with essentially the North water network or the potential transmission routes. And the mapping activities is so powerful and the processes involved are so powerful that it can allow us to actually track right down to GPS level. And we used this mapping to guide what particular locations were optimal for us to go and collect samples as part of our point prevalence and our longitudinal studies. And this mapping activity and this mapping process was published in 2019. So in the task 2, what we aim to do is collect samples in each of the 4 local authority areas and collect samples from hospital sewage, sewage of nursing homes, airport sewage, the influent and effluent from wastewater treatment plants, the influent and effluent from integrated constructed wetlands. We collected raw waters from drinking water treatment plants, the raw and treated waters from group water supplies, and we collected samples from estuaries, rivers, lakes, and seawaters. So we carried out a point prevalence survey initially, so carried out a wide range of sampling across the 4 local authority areas. We collected samples from each of the areas that I mentioned and collected 70 samples in total. And we detected ESBL producing Enterobacteriaceae, a particular type of antimicrobial resistant bacteria, in 39 of those 70 samples, and a CPE, a quite concerning type of antimicrobial resistant bacteria, in 9 of those samples. Interestingly to note, we just detected antimicrobial resistant organisms in the influent to wastewater treatment plants, and as you will hear, largely in the influent to integrated constructed wetlands. It's worthy of noting that when we look at the characteristics of the isolates we collected from water and sewage, there is a significant level of multi-drug resistance. So this is resistance to more than one type of antibiotic. And this includes resistance to some of the antibiotics that are very important for human healthcare. In task 3, we're looking at the role of land spreading in the dissemination of antimicrobial resistance into grasslands and soils. And we've collected DNA from the manures themselves, from the grassland and from the soil, and this has been analysed to see relationships. We also have assessed the ability of integrated constructed wetlands to remove antimicrobial resistant organisms from farm effluents. So we collected 96 samples, 48 influent and 48 effluent samples over a 1-year period from 4 farms. And we isolated 86 ESBL, so a particular, again, type of antimicrobial resistant bacteria, but interestingly, primarily from influent. So this suggests that integrated constructed wetlands have an impact on the removal of antimicrobial resistant organisms from that waste. We also evaluated the impact of anaerobic digestion on the removal of antimicrobial resistant organisms from manure. And we found, depending on the amount of total solids that was present in that manure, that on average, the CPE, so we spiked manures essentially with a particular type of antimicrobial resistant organisms. We passed it through the anaerobic digestion process, and we found on average, after about 7 days, the CPE were removed. And again, this work on the CPE removal using anaerobic digestion has been published earlier this year. So finally, we're developing a risk ranking protocol to assess the relative contribution of various sectors and the sources and levels of antimicrobial resistant organisms in the environment, and are developing health and agricultural models to achieve this. So overall, the AREST project will generate national level data on the key sources, hotspots and drivers of antimicrobial resistance in the environment from the healthcare and agricultural sectors, which will inform priority areas for action. We will provide evidence of the extent of contamination of the environment nationally with antimicrobial resistant organisms to again support policy decisions and engage better with the health, agricultural and local authority sectors on antimicrobial resistance. We will input the data generated back onto the initial maps to provide engaging visual representations of the data, and this will embed the One Health concept and build the capacity of Ireland's research community to support Ireland's National Action Plan on Antimicrobial Resistance. There is a lot still to do. There is a significant need for systematic surveillance and a harmonised approach to surveillance of the environment for antimicrobial residues and antimicrobial resistant organisms. We need to enhance our understanding of the impact of wastewater treatments. We've demonstrated potentially the impact of integrated constructed wetlands, but we need to look more closely at the level and types of different wastewater treatment processes in existence and how these impact on the removal of residues and antimicrobial resistant organisms. It's great to see that the EPA and HSE have jointly funded the AREST project, and there needs to be an enhanced One Health approach, bringing together the different sectors to funding research in this area. We need to develop better mechanisms to translate our research findings into policy and bring it to the policymakers in a more streamlined fashion. And we need to better enhance our communication of the concepts of One Health and also the issue and the important issue of antimicrobial resistance to the general public. So finally, I'd just like to thank my own research group at NUI Galway, the EPA and HSE for funding this research, and all of the project partners on the AREST team. I'd be happy to take questions, and if you have any questions, please place them in the app. Thank you.